The eye is a special sensory organ that gives rise to vision and a hollow, slightly elongated sphere formed by a three-layer wall. The internal structures are the sclera and cornea, which makes up the exterior layers. The uvea is the middle vascular layer subdivided into iris, ciliary body, and the choroid. The retina constitutes the innermost layer and it is made up of nervous tissue. The three layers are the fibrous tunic that comprises the sclera and cornea, vascular tunic that forms the iris, ciliary body, and the choroid, nervous tunic also known as the retina. Inside the eyeball, there is a zonular fibers also called suspensory ligament that divides the eye into two cavities, the anterior and posterior cavity. The three chambers of the eye are the anterior chamber that extends from the posterior surface of the cornea to the iris, posterior chamber that extends from iris to the front surface of the lens, vitreous chamber that extends from posterior surface of the lens to the retina. Aqueous humor is a watery fluid similar to plasma that fills the anterior and posterior chamber. The vitreous body is a translucent gel-like substance that also fills the vitreous chamber. The fibrous tunic has two parts, the translucent cornea which is in front of the eyeball and white in the sclera in the back. The cornea focuses incoming light into the retina and serves as protection in the eye.
The next is the sclera. The sclera is the white part of the fibrous connective tissues that covers the majority of the eyeball. The place where the cornea and sclera is joined is called the limbus. The sclera is made of connective tissues composed of bundles of collagens and elastic fibers. These fibers are less organized than in the cornea, and that contributes to opacity. The main cells in the scleral stroma are called fibroblasts. The next is the vascular tunic, also known as the uvea, is the layer of the eye situated between the outer fibrous tunic and the inner nervous tunic. The vascular tunic is comprised of three parts: the iris, the ciliary body, and the choroid. The iris is the anterior portion of the vascular tunic and is visible through the cornea. The iris divides the anterior cavity of the eye into the anterior and posterior chambers. The next is the ciliary body. The ciliary body is the part of the vascular tunic that is adjacent lens. It consists of the ring of ciliary process, finger-like projection of vascular tissues covered by the ciliary epithelium that secretes the aqueous humor which fills the anterior and posterior chambers of the eye. The next is the choroid. The choroid is the largest parts of vascular tunic extending from the ciliary body to the optic disc. Where the optic nerves leaves the eyeball, the choroid is made of capillaries and is responsible for the nutrition of the outer parts of the retina. As such, it is tightly attached to the underlying retinal pigments epithelium, but loosely attached to the overlying sclera. The next is the nervous tunic or the retina. Is the neuronal tissue that contains the rod and cones photoreceptors that sense and process light in the visible range. In the retina, the energy of visible light is changed into electrical impulses that travels to the brain and are intercepted by neurons in the visual context to provide vision. The rods and cones are photoreceptive cells located in the deepest layer of the retina that can convert visible light to electrical impulses. Both rods and cones have two segments: the outer segments, where the photoreceptors are located, and the inner segments, which is comprised of the cell body and the short axon. Rod cells are highly sensitive to light but provide minimal detail and contribute little to no color vision. Rods are prevalent in the outer areas of the retina. Rod cells are responsible for peripheral vision and night vision. Cone cells are function best in the bright light and provide detailed color, provide acute vision for activities where detail is essential. The outer segments of rods and cones form the photoreceptor outer segments layer. The inner segments of the rods and cones containing the cell bodies form the outer nuclear layer.
The type of tissue of cornea is composed of six layers epithelium, stratified squamous, Bowman's layer, stroma, collagen and keratocytes, descemet membrane, and endothelium, simple squamous. The function in the body is primary refractive surface of the eye, providing transparency and protection. The location is at the anterior part of the eye, covering the iris and pupil. The type of tissue of retina is a multilayered neural tissue. It includes ganglion cells, bipolar neurons, and photoreceptor cells, which are the rods and cones. The function in the body is to convert light into neural signals for visual processing. Lastly is the location in the body. It is found in the inner lining of the eye, between the choroid and vitreous humor.
Good day, Dr. Elena Olido, and to my blockmates. I, Christine Valenzuela, will discuss the histology of the ear, focusing on its importance, how sound is transmitted, the roles of key components, and the major parts of the ear. So let's start with why it's essential to understand the histology of the ear. First, hearing and balance function. Histology allows us to study critical structures like hair cells, the cochlea, and the vestibular system, which convert mechanical signals into nerve impulses for hearing and balance. Second, diagnosis of disorders. It helps identify issues like hair cell damage, which can lead to hearing loss, tinnitus, or balance disorders. Third one, treatment development. This knowledge is vital for creating therapies like cochlear implants and regenerative medicine to restore damaged sensory cells. The fourth and last one, clinical interventions. Understanding ear histology improves how we treat infections, trauma, or age related hearing loss, leading to better surgical outcomes. Let's summarize the three main parts of the ear and their functions. First, the external ear. It includes the auricle, auditory canal, and tympanic membrane. External ear captures sounds and protects the inner structures. Second, the middle ear. It composed of the ossicles and the eustachian tube. The middle ear amplifies sound and equalizes pressure between the ear and the environment. Third and last one is the inner ear. It includes the cochlea, semicircular canals, and vestibule. The inner ear converts sound into nerve signals, the cochlea, and regulates balance, the semicircular canals. So let's go into detail about how sound travels through the ear step by step. First step, sound waves enter the external ear and reach the tympanic membrane or the eardrum, causing it to vibrate. These vibrations match the frequency and intensity of the sound waves. Second step, the vibrations are transferred to the ossicles in the middle ear. First to the malleus, which is attached to the tympanic membrane, then to the incus, which acts as a bridge. Finally, to the stapes, the smallest bone in the body. These bones amplify the sound vibrations, increasing their energy to prepare for transmission into the fluid-filled cochlea. Third step, the stapes pushes against the oval window, a membrane connecting the middle ear to the cochlea in the inner ear. This action creates fluid waves in the cochlear chambers. Fourth step, these fluid waves travel through the cochlear duct, displacing its flexible membranes. The movement bends the hair cells in the organ of Corti, the sensory structure for hearing. As the hair cells bend, ion channels open, allowing ions like potassium to flow in. This creates an electrical signal, which is the first step in converting mechanical vibrations into nerve impulses. Fifth step, the electrical signal triggers the release of neurotransmitters, which activate the sensory neurons of the cochlear nerve, or the part of the vestibulocochlear nerve. These neurons generate action potentials, which are sent to the auditory cortex in the brain for sound perception. Sixth step, meanwhile, the excess energy from the fluid waves is dissipated into the tympanic duct and then exits to the round window, preventing echoing or damage in the cochlea. This intricate process allows us to perceive and interpret sounds with incredible precision. Here are some critical structures in the ear and their functions. Vestibulocochlear nerve, or the cranial nerve 8. It transmits hearing and balance signals to the brain. And the lymph, a fluid essential for stimulating sensory hair cells in hearing and balance. Tympanic membrane vibrates in response to sound, transferring energy to the middle ear bones. Last, the organ of Corti. Found in the cochlea, it converts sound vibrations into nerve signals for hearing. So let's dive deeper into the two types of hair cells in the cochlea and their crucial roles in hearing. Inner hair cells, or the IHCs. Inner hair cells are the primary sensory receptors for hearing. They are essential because they directly convert mechanical sound vibrations into electrical signals, which are then sent to the brain for interpretation. Without inner hair cells, we wouldn't be able to perceive sound. So when sound waves create fluid vibrations in the cochlea, these vibrations cause the basilar membrane to move. As it moves, the stereocilia, or the tiny hair-like projections on the inner hair cells, this bending opens ion channels, allowing positively charged ions to enter the cell. The influx of ions generates an electrical signal that triggers the release of neurotransmitters. These neurotransmitters activate the auditory nerve, which sends the signals to the brain's auditory cortex for processing. Essentially, the inner hair cells act as the bridge between the physical movement of sound and our brain's ability to understand it. Second, outer hair cells, or the OHCs. Outer hair cells are not the primary sensory cells, but play a critical role in enhancing hearing. They act as mechanical amplifiers, helping to fine tune and boost the sound signals. This ensures that we can detect even very soft sounds and distinguish between similar frequencies. So, outer hair cells are embedded in the basilar membrane and are influenced by the fluid waves in the cochlea. When these cells detect vibrations, first, they change their shape rapidly through a process called electromotility. This movement amplifies the vibration of the basilar membrane. This amplification increases the deflection of the inner hair cells, making it easier for the inner hair cells to generate electrical signals, especially for faint sounds. Additionally, outer hair cells help improve the clarity of sound by suppressing background noise and sharpening frequency resolution. This is why they are sometimes referred to as the cochlea's fine tuning system. In summary, inner hair cells are responsible for transmitting sound information to the brain, while outer hair cells enhance and refine this process, ensuring precise and sensitive hearing. And that's all.
The type of tissue of Corti. The organ of Corti is composed of specialized epithelial and then sensory cells, including inner and outer hair cells, supported by various types of supporting cells like Dayter cells, Hensen cells, and pillar cells. The function in the body, this structure is critical for auditory transduction. It converts mechanical sound vibrations into electrical signals that can be interpreted by the brain as sound. The hair cells, sterocilia, respond to fluid displacement in the cochlear duct, activating the auditory nerve. Lastly is the location in the body, dislocated within the cochlear duct of the cochlea in the inner ear, specifically on the basilar membrane. This region is part of the auditory system responsible for detecting sound frequencies. The type of tissue of foliate papillae is a stratified squamous epithelium with taste buds housed in crypts. The function is to sensory reception of taste. Lastly is the location in the body. It's located at the posterior lateral aspect of the tongue. The type of tissue of olfactory tissue is a pseudostratified columnar epithelium with specialized olfactory receptor neurons, sustentacular cells, and basal cells. The function in the body is to detect odors and transmit signals to the brain. The location is found at the roof of the nasal cavity, near the cribriform plate of the ethmoid bone.
The surface of the tongue, mostly the dorsum, is covered with mucosal elevations that facilitates the uptake and sensory assessment of food, protect the tongue by providing a rough surface for cleaning the oral cavity, assist in the mechanical breakdown of food, enhancing digestion, detect textures and temperatures, and also the chemical properties of substances for sensory feedback. Term the lingual papillae. These structures vary in their size, number, and distributions over the surface of the tongue. A distinction is made between mechanical papillae and gustatory papillae, which are further subdivided as follows. For the mechanical papillae or the papillae mechanicae, which includes the piliform papillae or the papillae filiformis, the conical papillae or the papillae conicae, and the marginal papillae or the papillae marginalis. And for the gustatory papillae or the papillae gustatoriae, which includes the fungiform papillae or the papillae fungiformis or the valate papillae or the papillae valatae, and the foliate papillae or the papillae foliatae. The mechanical papillae typically have a connective tissue core housing the numerous nerve fibers and mechanoreceptors. Tactile stimuli are received by free nerve endings in the epithelium, and the papillae are supplied by a dense vascular network. The types and characteristics of mechanical papillae. The filiform papillae of horses, pigs, and goats manifest as soft threads of epithelium that give the surface of the tongue a velvet-like smoothness. In cattle, sheep, and cats, small keratinized spines project towards the pharynx from a connective tissue core. These are responsible for the characteristic roughness of the surface of the tongue in these species. In cats, the protruding spines are supported on the rostral aspect by an additional papilla. For the blunt conical papillae are found in the carnivores, pigs, cattle, and small ruminants. In neonatal carnivores and piglets, the margins of the tongue bear elongated marginal papillae. These form part of the lateral boundary of the oral fissure and facilitate suckling. In gustatory papillae, the stratified squamous epithelium contains specialized structures, termed taste buds, that are concerned with the perceptions of taste stimuli and detect and differentiate basic taste sensations such as sweet, sour, salty, bitter, and umami. Enhance the appetite and regulate food intake by evaluating flavor and palatability. House taste buds containing sensory cells that transmit taste signals to the brain. Contribute to oral health by triggering salivation, which aids in digestion and cleansing the mouth. Provide sensory feedback about the chemical compositions of some substances in the oral cavity. The types and characteristics of gustatory papillae. Fungiform papillae are distributed over the dorsum, margins, and parts of the ventral surface of the tongue. They appear as a small mushroom-shaped elevation that protrudes slightly above the tongue surface. Fungiform papillae are not as strongly keratinized as mechanical papillae. It is thought that fungiform papillae may contain thermoreceptors. Functional taste buds are present in the epithelium in calves and horses. In carnivores, they are abundant; taste buds are more numerous in young animals during the suckling period. For the valate papillae, occurs at the transition from the body to the root of the tongue. Horses and pigs have one pair of valate papillae; two to three pairs are present in carnivores. Valate papillae are most numerous in ruminants, and each papilla is surrounded by an annular sulcus, the outer boundary of which is slightly elevated. The papilla itself does not protrude beyond the surface of the tongue. The sulcus is lined by the epithelium. On the papillary side, the epithelium contains numerous taste buds, which is the number varies with species. The tubuloacinar serous glands, or the Ebner's glands, gustatory glands, lie deep to the valate papillae, between the tela submucosa and muscle fiber bundles. Their secretory product passes through a branch system of ducts into the base of the sulcus. Continuous production of secretion ensures that dissolved nutrients within the saliva are constantly washed from the surface of the taste buds, allowing the gustatory sensation to be detected. Lastly, for the foliate papillae, are located laterally near the root of the tongue. They are well developed in the horse, less so in pigs and carnivores, and absent in ruminants. This gustatory papillae consists of folds of mucous membrane separated by transverse furrows. The walls of the furrows house numerous taste buds. Dense accumulations of gustatory glands similar in structure and function to those associated with the valate papillae are present beneath the furrows. Gustation is mediated by chemoreceptors located within the taste buds, or the Jamie gustatoriae. Taste buds are found primarily on the surface of the tongue and in the pharynx. They are abundant on the side walls of the valate, foliate, and in young animals, fungiform papillae. Taste buds contain secondary sensory cells that develop by modifications of epithelial cells. Taste bud structures and function. The size of a taste bud is usually 50 to 70 micrometer, consists of intraepithelial clusters of 20 to 30 individual cells. They extend from the basal lamina to a small taste pore at the surface of the epithelium. Within the taste pore, these substances stimulate chemoreceptors on sensory cells in the taste bud, numerous up to 50. Free nerve endings located at the basolateral aspect of the taste bud direct the stimulus to the central nervous system. The lifespan of taste bud cells is approximately 10 days. They are replaced by basal cells. Cell types in taste buds. The pillar-like sensory cells or the chemoreceptor cells are situated between sustentacular cells. Microvilli extending from the luminal surface of the sensory cells serve as the site of excitation. Basolaterally, the receptor cells synapse with axons that constitute with the initial fibers of the gustatory neural pathway. Sensory cells can be distinguished with the light microscope. They are rich in organelles, and for the supporting cells, also extend through the full thickness of the epithelium. This contains abundant enzyme-forming organelles and dense endoplasmic reticulum. Glycosaminoglycans synthesized by supporting cells are released into the taste pore to facilitate taste perceptions. Supporting cells are thought to be capable of differentiating into sensory cells. And for the basal cells are differentiated cells that undergo mitotic division to provide continuous replacement of sensory and supporting cells. They have relatively few organelles and are situated around the nerve fibers. In some regions, the lamina propria mucosae contains serous tubuloacinar glands or the gustatory glands. The secretory product empties via excretory ducts into the sulcus of volate papillae and sometimes into the spaces between folate papillae. The watery secretion rinses the taste buds, permitting the detections of new stimuli. The number of glands increases with the number of taste buds.
The next is the sense of smell or olfaction. The sense of smell occurs when odorants binds to olfactory receptors located in the nasal cavity. There are many types of odors receptors, and each respond not to a single odor but to a number of similarly structured odorants. This diversity allows for a million of different smell, even the smell never before encountered, to be discerned. Signals from the odor receptors are sent to the olfactory bulb in the brain via the olfactory nerve or cranial nerve 1, where they are interpreted as smell. The olfaction is not only tied to memory and emotion but combines with taste from the sense of flavors in food.